Okay, so I'm continuing discussion of the processes of isolation and control experiments with respect to microentities, real or supposed, such as viruses. And I have to be careful what I say. I had a post recently which was taken off YouTube because I stated certain facts which they don't like to hear. So to avoid similar outcome, I'll just keep it to the process in the abstract. I was actually asked for some sort of diagrammatic representation of what I was saying, so this is my humble offering. So, looking at the first picture, this indicates some sort of liquid which is causing some sort of damage. That's how I've represented it. You can see the full length lines there. The liquid falls on it, and some of the lines shrivel up. Let's say they become half size. We could say that as a picture of cell damage. Let's say some sort of cytopathic effect is the technical word for cell damage, is caused by this liquid. So that's the problem. The question is, what is causing the damage? Okay, so looking at that at a more detailed level, at a scientific analysis level, we have a test tube which contains the liquid, and you can see the liquid has various elements represented by dots, circles, and plus signs. Of course, a real liquid would have many more different elements, but that just shows you the basic point. And the idea of isolation is that you somehow produce only the matter, the substance, the entity that you think is causing the problem. So that's what isolation is, because, as I said in the last video, you have to show that X causes Y. If you want to show X causes Y, you have to have X by itself. That's what isolation means. If you have X, Y, and Z together in the mixture and they produce the effect, well, all you've shown is X, Y, and Z produce that effect. So what we need is this process of isolation. The question mark represents that there's some method of producing only the entity which is said to cause the problem. Of course, in the great issue of our time, the so-called virus, and you need to produce that in a pure form. Okay, so what we would be looking for in a scientific paper is saying, okay, we needed to produce the virus, to isolate it, so we just have the virus, and we did this process, ABC process, whatever it was, and we achieved pure virus and nothing else. So that's the question. I'm just going to ask the question. I'm not going to make any statements as to what I think the answer is. But if you're looking at a paper, does it endeavor to do this? Simply that. Does it say, well, we wanted to achieve this, and this is how we did it, and we had pure virus? So that's the question I want to ask. I want you to ask about this or any other virus is, has that been done, or have the people at least said, well, this is what we're trying to do, this is what we try to do? That's isolation. It's just a fundamental common sense thing that if you're saying something causes an effect, then you need to get that thing by itself and produce that effect with it. And that's what figure C really shows you. You have the isolated substance, and it causes that cell damage. It's established that basically that part of the substance causes that damage. You haven't actually established that the other parts don't cause it. Actually, you'd have to do other isolation processes. If we have these dot, circle, plus sign, you'd have to do a similar process for each of those in order to see exactly which one or which combination is causing the problem. So that's isolation. And figures D and E represent control experiment. So this is another one you need to be looking at if you're examining a paper about the isolation of the virus. Was a control experiment, or were control experiments carried out? And the point is, so you're taking a sample of cells, we're saying these lines are cells, and in reality, of course, if you want to see if there's been cell damage, you need some sort of process to make it microscopically visible. And so what the next part of that image represents is putting some preparations onto the cells in order to preserve them so you can view them. Okay, so that is a process in itself of preparing the slides, preparing the visible culture that is going to be tested. And in the main experiment, D, you would then add the isolated virus, if you actually had that, and put it onto the cells, and then you would see the damage that is the process that is claimed to show that virus causes the problem. But what you also need is a control experiment. So again, without making any statements, I'm just asking you, have you seen a paper which says yes? We also did a control experiment whereby we just went through stage one. We just prepared the cells. And what happened then? And you look, are they damaged, as in the first part, or undamaged in the second? So that is important, because you can only say that the isolated substance that you see there in D is causing the problem, causing the damage to the cells, if the process of preparation is shown not to cause the problem. It doesn't cause damage if it's just done by itself. Okay, so without stating what has happened when this has been tried or whether it's been tried or not, I'm just saying that these are the two important concepts: isolation, that's represented by ABC, and control experiment represented by DE. And that's it. I just hope that's as simple, just to understand clear concepts. We need to apply common sense and logic to this and just follow these things. We have God-given reason. And something like this is the only way to show that something you can't see. I mean, if you can't see something in an obvious way causing a problem, but at this very small level, something like this as a way of establishing that what we're claiming is the cause of a problem actually is that. So I just ask people to think about these things and follow the consequences of what they find. Thank you very much. Oh yes, and I should just add that the link for the original video is in the description below.